we're going to do brain today right after we get into, we're going to do blood-brain barrier first. I'm going to tell you about this cool app that exists. It's free, okay? And it's called 3D Brain. I know it is available on iOS devices. It's really, really good. It's very good quality. It was put together by Cold Spring Harbor Laboratory, which is one of the best research laboratories in the country for biology, that is. It's free. It was funded by the federal government as part of the Human Genome Project. Some of us are old enough to remember where the Human Genome Project was going on, and some of you guys, it's always been that. Yesterday. So as part of the Human Genome Project, they had to do an education component. And this is part of what came out of the education component. So they can't charge for it because you already paid for it with your tax dollars, or your mama did. But if you access it through a computer, here's where you find it. So they also have a web-based version, which is G2C online. We'll use it today. So let me show you where you would find it. So watch, okay? Here you go. So Genes to Cognition online has tons of information about a whole bunch of neurological stuff. But the portion that we use in anatomy is right here with 3D Brain, right there. And we'll use that today as we're going through the brain. It's especially good for people like me who are very, very weak in spatial organization, because it's super hard to figure out where structures are of the brain relative to one another. For me. For some of you guys, you get it really quickly, but for me, it's really hard. So we're going to refer to this several times. I won't test off of it because I don't know how to test off of it, but I think it helps to reinforce the stuff that you're learning. Okay, so that's first. Is it available on Android? Does anybody have an Android device that could look? No?
Oh, well, I got it on Samsung.
Oh, is it on there? 3D Brain?
Yeah.
Okay, cool. Yeah, it's fun. Your kids will like it too. Okay, so before we get into the brain structure specifically, we have to finish up with blood-brain barrier, which was the last line of protection. Okay, so remind me what the first lines of protection were. What was the very first line of protection of the central nervous system? Cranium, the bone. Next ones, collectively? Meninges, good. Next one? Good, cerebrospinal fluid. And then now we are to the blood-brain barrier, okay? So we have all of these lines of protection. But I need to explain to you both about blood flow to and from the brain, and then also what I mean by blood-brain barrier before we get into the specifics. First off, the brain is a blood hog. It only accounts for about 2% of your body weight, but it uses up about 20% of your glucose and oxygen. So it requires a ridiculous amount of blood flow. And in addition, it is intolerant of, I don't know if you know the word ischemia. Do you know that word? It's reduced blood flow. It's intolerant of reduced blood flow. It's in your notes right here. So it's intolerant of ischemia. It will die at average temperatures with about a 1 to 2 minute interruption in blood flow. So if you're going to drown, for instance, and not give oxygen to your brain, make sure you do it in cold water because it also lowers the metabolic needs. Right? But try not to drown. So it uses up 20% of oxygen, 20% of glucose. It has almost zero capacity to perform anaerobic cellular respiration. What do I mean by that? Make ATP in the absence of oxygen. So basically your brain without oxygen cannot do much of anything. So your brain needs a whole bunch of blood flow, and it gets this blood flow primarily via 2 pairs of vessels. I'm not going to test you on the names of the vessels yet. I'm just going to tell them to you now so that when we learn them in detail in the cardiovascular system, you'll be able to put it together with the story that we already told, okay? So your brain gets blood flow through 2 pairs of arteries. Let me show you where they are. These 2 right here are called the internal carotid arteries, and they come up at the base of the brain through holes that you know. Carotid canals, exactly. Do you see these? They're cut right there. They come up right there. The pituitary gland would be hanging off right there. What did you say? Oh, this is anterior, and this is posterior. Got it? Everybody okay? Okay, so internal carotid arteries, 1, 2, right there. Those 2 collectively do about 75% of the blood supply to the brain, the pair of them, okay? And I know it's not an even number, just stick with it, okay? And then these 2 right here are coming up just anterior to the medulla oblongata, right? So this would be connected to your spinal cord, or oriented. So those are called the vertebral arteries because of this. Because when they are running up, they actually run through those side holes in the cervical vertebra. What are those side holes called? Transverse foramina. They run up the transverse foramina, and then they fold anterior to the medulla oblongata, and then they run parallel up the foramen magnum. Yeah? Okay. You have 4 of them, 4 total. Okay. Why do you think the redundancy is a fantastic evolutionary plan? Yeah. And I am an example of how this keeps you alive, because my right vertebral artery, about 6 or 7 years ago, busted a hole in it. Just like, whoosh. And then all of the blood that was supposed to be going up there just dumped directly into my venous system. Should have probably had a stroke or died, but because of the redundancy of the system, I did not. Maybe I used to be smarter. I don't know, because this is as smart as I am, right? And I don't know how smart I used to be, and you can never tell, right? So I don't know. So I'll tell you guys more about that when we get into the cardiovascular system. But I just wanted to tell you, basically 4 blood vessels is keeping your brain alive. Now, once you get in, and I want you to picture this, because I don't know, some of you guys had anatomy in high school and some of you didn't. So if I'm over-explaining, it's not because I think you're stupid. It's just because anatomy is not required. That's all. So arteries, of course, are going to split to become smaller arteries, yes. And then they're going to split to become smaller arterioles, right? And then they're going to split to give blood supply to the entire brain via almost a complete capillary network that completely suffuses the brain. Every single neuron is within millimeters of a capillary. Yeah? So it's really, 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 really vascular tissue. Got that picture? Okay. So I wish it was darker in here, but I was too lazy to close those. So I was like, I want it to be darker, but no. Okay. So this is all of the blood. This is the whole head. Okay. So you know this. You are going to, of course, be bringing oxygen and nutrients and picking up carbon dioxide and waste products. Yes, like you do at every single capillary. And then once you get done, you already know what's going to happen. You're going to dump it back into these big old vessels that are going to drain the blood from the brain. What are those collectively called? 3 words. One of them starts with the duh. Dural venous sinuses, yeah? And the dural venous sinuses are all going to gather again, and they are going to dump into the internal jugular vein that goes through a hole that you know, jugular foramen, right? And then you realize that this is all one damn class. It's not 72 different classes. It's all one class, right? All coming together, right? Jugular foramen. Yes? Okay, so, and I've got a cool picture for you. I forgot that I had this last time. Look at this cool ass picture. That's a dissection showing you all of the dural venous sinuses. I love it. It's so nice. And then this one is showing you all of the blood vessels going into the capillaries of the cerebral cortex, which is pretty damn cool. Okay, so now what we need to get to is, what is the blood-brain barrier? Well, what I want you to picture is take one of these capillaries that's deep within the brain, and I want to talk about how that capillary can protect the brain tissue that is around it. Okay? It can both supply what the brain tissue needs and also protect the brain tissue from any potential damage most of the time, and it works pretty well. So let's talk about that. Okay, so if you look at one of those capillaries, it has what we call a blood-brain barrier. And the blood-brain barrier is really 2 things. It's both an anatomical barrier and a physiological barrier. So what I want you guys to be, you guys are all neurons in the gray matter, and you're very, very important. Your mom told me. And these guys are the capillary cells, right? Okay? And the capillary cells are all stuck together via the tightest of tight junctions, yes? And I am a toxin, and I want to whoop up on you, right? But they are not going to let me through because they are stuck together with the tightest of tight junctions, which is they are called continuous, complete tight junctions. Basically, I am not messing around tight junctions, okay? So that means that if I'm a toxin or a pathogen circulating in the bloodstream and I try to get through, there is no way I can get between the cells, yes? And then I ask permission. Can I come in? And he says, hell no, right? You are not welcome here. Does that make sense? Okay. So it provides a tremendous amount of protection just because it provides a relatively impenetrable barrier between the cells. Relatively impenetrable because if you're lipid soluble, Are you guys going to be able to protect them from me if I'm lipid soluble? Lipid soluble stuff does whatever the heck it wants, right? If I'm a lipid soluble toxin or pathogen, you can't protect from me because, of course, you're made of lipids, right? But if I'm any other kind of thing, they can protect. So that's how it's an anatomical barrier. And then the second way it's a barrier is it's a physiological barrier because the astrocytes that help form the blood-brain barrier, they don't just like form a physical barrier. They also secrete chemicals that can change transport rates based on what you guys need. So if you are working your little neuron butts off, thinking as hard as you can about anatomy, and you need more glucose, what the astrocytes can do is secrete chemicals that increase the glucose transport rate into the brain. Yeah? If you are producing too many waste products, they can secrete chemicals that increase the transport out, right? What if something funky is going by? The astrocytes can secrete chemicals that shut it all down, right? So that it won't get in. So it's both a physical barrier because of the tight junctions and a physiological barrier, right? Because it can change the rate of transport. For those of you who have had phys, the way that it does it is primarily by releasing paracrinagens that change the transport rate of like glucose transporters or amino acid transporters or whatever. Does that make sense? Okay, so it's a physical and a physiological barrier. So so the astrocytes do it. So how does stuff get in? Well, stuff gets in because you have a transport mechanism for it. So I can't go between you, but I could go through you if I were glucose and you had glucose transporter on you, right? So if it's stuff that you need, like glucose and amino acids, there are going to be transporters on the capillaries to get it in. Other substances don't have to ask permission. Oxygen is lipid soluble. It diffuses straight in down its concentration gradient. Other things are lipid soluble, and sometimes that's good and sometimes it's bad. Nicotine is lipid soluble. That's one of the reasons why you can feel it in the brain really, really quickly. Alcohol is lipid soluble. That's one of the reasons you can feel it in the brain really, really quickly as well. So alcohol is lipid soluble. So if you didn't have dinner and you had a sip of wine, right? Because you were trying to be efficient, right? You have a sip of wine, right? No reason to waste any money, right? Have a sip of wine, or calories, exactly. You're like, I'm drinking dinner tonight. So you have a sip of wine. How long will it be before you feel that in your brain?
2 minutes.
Yeah, within a matter of 30 seconds to 2 minutes, depending, right? Because it's lipid soluble, it actually diffuses straight into your brain. Alcohol diffuses straight into your brain. When you take physiology, I will teach you what it does to your brain, like how it actually works, what it changes about the physiology of your brain, because I talk about that stuff a lot. So anesthetics are also lipid soluble. That's one of the reasons they work really well, but we don't usually inject them into the vessels, right? Anesthetics are usually localized. They're either regional or they're injected into the epidural space, but not into a blood vessel, right? Even if you did inject them into a blood vessel, they would diffuse into your brain, but it won't make any difference because you don't have any sensory receptors in your brain. So you wouldn't be able to feel it that it happened or feel it that you deadened it, right? You have no sensory receptors in your brain. That's why they can do like living brain surgery. Have you guys seen brain surgery like on a living person and they're still talking or playing an instrument or whatever? Okay, now, other substances, if there is not a transport mechanism for them and they're not lipid soluble, they're probably not getting in. Sometimes that's good because it's something weird. Sometimes it's bad because it's an antibiotic and you need it in there, right? But it will protect you from from anything without a transport mechanism or lipid solubility. So here's an instance in which it's an impediment. So with Parkinson's disease, because we talked about before, your glial cells stop supporting your neurons. The neurons stop being able to produce a neurotransmitter called dopamine that in that region of the brain controls voluntary motor activity. So that's a problem. We don't have dopamine in an area of the brain that we know. It's called the substantia nigra. You need some dopamine. Here's what we do know. We know how to make dopamine in the lab and we know where it needs to go. But we can't get it in there because it's not lipid soluble and there's no transporter mechanism for it, right? So sometimes it's good, sometimes it's bad. That workaround that has been going on for, 10 or 20 years is that in the early stages of Parkinson's, you can give the lipid-soluble precursor to dopamine called L-DOPA. And that diffuses in, and as long as there are still enough cells to do the conversion of L-DOPA to dopamine, you get a relief of the symptoms. Yeah? But then in the later stages, it stops working again. Does that make sense? Because you don't have enough living cells to do the conversion. Is that okay? Okay, so that's with Parkinson's disease. The other thing that the blood-brain barrier does is it kind of buffers you guys against extracellular fluid or blood vessel fluctuations. Like after I ate, my sugars go really high, but you guys stay relatively normal because these guys will slow down in glucose transport, right? When I'm sleeping, my blood sugar goes really low, but you guys still need almost as much sugar as you ever did, so you just increase the transport rate, right? So really what it's doing is buffering the central nervous system's experience, but I don't just mean pH buffering. I mean like metaphorical buffering, right? It's like evening out what's happening with the brain so that it doesn't really experience really big extracellular fluid fluctuations, right? So it's doing that. Of course, it also protects the brain from pathogens. Now, if there is anything good to say, and there's hardly anything good to say about smashing yourself in the head, it is that if you have inflammation in the brain from like a traumatic brain injury or from a concussion, it temporarily destroys the blood-brain barrier, and the only good thing about that is that you can get drugs into the brain that you couldn't otherwise get into the brain. So if you have a head wound, the first thing they will be concerned about is swelling, right? So generally speaking, they'll give you a really, really powerful anti-inflammatory, usually like prednisone or a cortisol derivative, like we talked about in class yesterday, right? Cortisol derivatives. So they'll give you one of those as a massive anti-inflammatory, but then they'll also give you an antibiotic just in case, right? Just in case something got in there, right? They call them prophylactic antibiotics, not because they're shaped like condoms, just because it's like, hey, this is protective. So they're prophylactic antibiotics that they will actually give to you. Have you guys heard that term before? Prophylactic antibiotics? Okay. So steroids and antibiotics. And here's one cool thing. So this, I think, just happened either last year or the year before. Some clever person came up with a fantastic way to get temporarily around the blood-brain barrier and allow targeted delivery of drugs to places in the brain. Like, for instance, if I have a tumor in a particular portion of my brain, I know exactly where it is, and I have a chemotherapy drug that I need to get targeted to there, but I don't need it to go generally to the brain because that stuff is rough, right? I need it to go to one little area, and it's not lipid soluble. How the hell do I get it in there? Some brilliant freaking person, last year or the year before, I wish I could remember when, I forgot to write it down, came up with this thing. So they figured out that if you inject these microscopic gas bubbles into the bloodstream, not big enough that it could cause an aneurysm or anything. We're talking like smaller than carbonation bubbles, okay? Teeny tiny little gas bubbles that you inject into the bloodstream, right? And they're circulating around the bloodstream and they're really not doing much of anything to anywhere. But then if you take those microscopic bubbles and you blast them with ultrasound at a specific location, like where the tumor is, what it will do is temporarily destroy the blood-brain barrier, yeah? And then you inject something that is not lipid soluble and is a chemotherapy drug into the bloodstream. And then where does it get in? Only at that little location right there where the blood-brain barrier was destroyed. And then guess what happens as soon as you stop zapping it with ultrasound? It closes right back up. Freaking brilliant, right? Right? Like every brilliant idea, well, almost every brilliant idea outside of physics, it sounds simple, right? When you actually see it, you're like, oh, that makes perfect sense. That's great, right? Outside of physics, because I can't even understand the answers even, or the questions half the time, and especially like theoretical physics. Have you guys ever read any of that stuff? Good way to make you feel like a freaking doorknob. Like you are apparently the dumbest person that's ever existed. Me, not you. Because I was like, I don't even understand what that sentence said. I don't know what you're talking about. Okay, so this is an experimental therapy. Last time I checked, I haven't checked recently, this was still in animal models in clinical trials. I don't know if you guys know how clinical trials work. Here in the United States, and perhaps everywhere, I don't know. Write a proposal, got to get a whole bunch of money. People say, hey, that's a great proposal. Here's some money. And then you're going to have to do, if it's going to ultimately deal with human subjects, you have to do in vitro testing, in which you like work in a lab with cells that are not part of a human and may not even be human cells, right? And then if that goes well, you use animal models, yeah? Quite often mammalian models like rats or things like that. And if that goes well, you can move it to human trials, right? With something like this, because it's potentially invasive with the central nervous system, you would have to have a person who was a good treatment candidate and was also not a whole lot of other choices, right? But also still had the wherewithal to say, I am willing to take part in this experimental trial, right? And I don't know if we are at the human trial stage yet of this. I haven't checked this year. So if you guys know, you can tell me. Okay, go ahead.
How do you follow them? Like, how do you know these? How did you know that? How could I follow this?
How could you follow this? Oh, the name of the treatment?
I have a friend that has that.
Will you email me that so that I can remember what it's called? Because I don't remember what it's called, but I can look it up. Okay. I thought you meant how do I find out weird shit like this? And I was like, well, it's literally all my brain does. Is like search, gather. Have you guys ever taken a personality test and they tell you what your personality predisposition is? I am a facts curator. Like, my personality loves to gather and categorize things and gather and categorize things and gather and categorize things. I love doing that. So that's why I know this stuff, because I never get finished gathering facts. It's the most entertaining thing in the world. My husband, we went on vacation, and my husband's like, what the hell are you doing? I'm like, I'm working on my neurotransmitter spreadsheet. He's like, why are you doing that? I'm like, it's because I love it. That's why I'm doing it. And he's like, there's a beach. And I was like, it's still going to be there when I get done. Okay. So now I want to talk about the organization of the brain. You guys ready? Okay. So I get super nerdy about the brain. It's literally my favorite thing. And I feel like I'm never, ever going to be finished learning about it. So the stuff that I'm telling you is the best knowledge that I have now. It's more than your textbook has. Okay, so first I want to give you the big picture understanding of the brain and tell you that, let's look at this picture for just a second. So one of the reasons that I like this is that it allows you to have a 3D understanding of where the brain is. And is it Seyon? Okay, Seyon asked me, like, anterior, posterior. Since you guys may or may not know, can you tell anterior from posterior yet in this figure? Okay, so this is anterior, this is posterior, this is superior, that's inferior, right? So we can flip it around and we can see all of that. And it's not hard when you're looking at a whole brain to tell, but sometimes when you start looking at the parts, it gets a little hard. And then you can view the labels too. The other thing that I like is that, of course, it doesn't just do the whole brain, it does things that are really hard to see, like let's find out where, later on, you guys will say, I don't understand where the hell the thalamus is, but now you will understand where the hell the thalamus is with reference to other structures, right? And you can flip it around and you can actually see it. The other thing that's fantastic about both the program and this thing is that for those of you who might be facts curators, does anybody feel like that sounds like their personality? Yeah, I was thinking she might be that kind of person too. So it has not just the overview, it's also got case studies, which is often how we learned what that part does. It's got associated functions, and then, oh my goodness, look at this. It's got clickable, peer-reviewed literature on individual portions of the brain, so you can go read the original literature, which to me sounds like angels singing. It's like the best thing ever. You might never read it, and that's totally fine. But this is why I have 3 friends, and they all know I'm super weird, and they're like, you probably shouldn't talk about that at dinner. And I was like, maybe you should have dinner with somebody else. Okay. Okay, so this is fantastic. So I'm going to use this a few times as we go through today, and you guys can use it if you've got it on your apps too. So I want to talk about the way the brain develops, both evolutionarily and also embryologically, okay? So this is kind of really important to understand. I'll give you a big picture understanding. Because when we're talking about the human brain, really all we're talking about is the vertebrate brain, right? With some extra functionality on top, just a little bit of extra functionality. So let's talk about evolutionary development of the brain, and then we'll talk about embryological development of the brain. The thing that I want you to understand about the brain is the oldest portions of the brain, both evolutionarily and also the ones that form like in the first trimester, are these deeper portion of the brain way down here. This right down here, this is the oldest portion of the brain. It's oldest both in functionality, I'll tell you the functions in just a second. It's also oldest evolutionarily, meaning you have it in common with not just a monkey, but your dog, your cat, a salamander, and fish, all of them, right? Okay. And it is related, the functionality of those old portions of the brain, including the brainstem, the medulla oblongata, things you've probably heard of, are life-sustaining functionalities like controlling respiration and controlling heart rate, right? So you have that in common with all of those critters, right? To greater and lesser degrees, right? Everybody has like at least some capacity to control that. So do you think that has anything to do with consciousness, those super old portion of the brain? No, nowhere near the consciousness level. You guys with me? Okay, so now we're going to move up to some newer portions of the brain, not the newest yet. These middle portions of the brain, which people usually are just not familiar with, in part because you can't see them from the outside, and even if you live kind of mid-sagittal section, you can't see them, right? So it's really hard to figure out what the hell you're talking about when you're looking at these functions. These are not life-sustaining functions, but they are, I don't know, this is an oversimplification, but they're like species-sustaining functions. Like sex drive is in there, rage and territoriality and repetitive behaviors, and like seeking behaviors and hunger and homeostasis and thermoregulation. And those are in there, right, in the middle portion. And you have those in common with most vertebrates as well, right, to greater and lesser degrees, right? As you get to higher vertebrates, you're going to have more behaviors that you see in common with humans. But obviously every sexually reproducing organism has something related to sex drive in there, right? Okay, so those are sort of, I think of them as kind of species sustaining, right? So life sustaining or individual sustaining and then species sustaining. And then the newest portion of the brain is the portion that everybody's used to looking at, which is the cerebrum. And it is newest in two ways again. It's newest evolutionarily, meaning that your cerebrum is going to be most similar to mammalian cerebrums. Yeah? I wonder if that's plural, I don't even know. And also it's the last to develop embryologically. So as you're putting together a fetus, right, first thing to develop would be the brainstem, the second would be the middle portion of the brain called the diencephalon and the mesencephalon in the middle, and then the very last portion to form would be the cerebrum, which is the outer portion. Now the cerebrum in us is really a mammalian cerebrum with more stuff on the surface. Okay, so this is an oversimplification too, but I do want you to kind of understand the difference. So I know that probably what you guys have heard before, and it's what I heard for a really long time, was that really the special thing about humans is that our brain is really, really big. That's not actually what's special. It's a little bit of an oversimplification. So let me show you some brains and talk about why our cerebrum, and specifically I'm talking about the cerebrum. So why is our cerebrum special? So these are a bunch of different cerebrums from a bunch of different critters, all mammals, I think. Yeah. I don't know what the hell a mouflon is, but it's probably a mammal. The real difference between humans and other mammals is just a matter of degree. It's really a spectrum of what we have here. So we can see that our brain is rather large, right? But so is a giraffe's brain. Really, there's two things that end up making us relatively special. Is that the cerebrum is primarily related to a couple of different things. It's related to conscious awareness of the sensations that you're experiencing. You guys with me? Okay. So your conscious awareness, voluntary control of motor activity, and all of those critters have all of that, right? Okay. Plus, if you still have a whole bunch of real estate left, you might be able to cause global warming, have an anxiety attack, or write a poem. Okay. So what the real difference is with us is the surface area of our cerebral cortex, the surface portion of the cortex, is really our processor, our processing capacity. And the surface area of the cerebral cortex, the most important thing is, yeah, your brain is big, but how big is your cerebral cortex relative to your body size? Okay, so what I want you to do is compare me to a giraffe and see what ends up happening there that makes us rather different than a giraffe, okay? So when you are a giraffe, you're giraffe size, and their brain is almost the same size as ours, right? Okay? But what I want you to understand is what they are going to have to do before they can do anything else is map every single sensation in a giraffe-sized body to some surface area, real estate, in the cerebral cortex. Yes? With me? And then they have to match motor control, voluntary motor control, to all of the musculature in a giraffe's body. And then see how much surface area you have left. Not very much for a giraffe, right? Because really the real estate is mostly taken up. And now let's do the same thing with me. I'm going to map all of the sensory activity for my body to real estate in my cerebral cortex. All of the voluntary motor control of my body to real estate in the cerebral cortex, and then see what I have left. And what I have left is a pretty distinctly large amount of cerebral cortex. Yes? And that large amount of cerebral cortex can be used to write a poem, have an anxiety attack, cause global warming, whatever the hell you want it to use for. Go ahead.
I don't know if this was just proven or not, but like the whole, we only use like 10% of our brain and we're not at full capacity.
No, it's not actually true. It's truthy. You know what I mean? Like anytime you see a soundbite about science on the internet, it's usually like just kind of a little bit of the truth. What is true is, generally speaking, our cerebral cortex has more capacity than we are using it for at any given time. But it's not as if it's just sitting there waiting. It's like when you're sitting around watching Friends for the 16th time, you got a lot of space that you could be doing something with. But is it not used generally? No, that's not the case. But you do also have a lot of what we call neuroplasticity, meaning that if I had a stroke and this portion was taken up, my processor is kind of all-purpose or general purpose, so I can pass that functionality off to another portion. But can we be doing more with our brain? Yes. The 10% thing isn't true, but the you can do more than you do with your brain is true. And we know that, right? You've been a human long enough that you know that you can do more with your brain. I mean, we don't always choose to do it because it's exhausting and sometimes it makes you feel like an idiot and we don't like to. That's why I don't talk about physics, because I'm like, okay, so it's kind of true. So, truthy. Just like the whole left brain, right brain thing. All I can tell you about that is that's just truthy, right? A little bit of truth, and you made it into a meme that looks good on the internet, right? But it's not like true enough for anybody to use it. Okay, so do you kind of understand? So the real difference between you and another mammal is both the size of your brain, but even more importantly, the surface area of your cerebral cortex relative to your body size. That's the biggie, okay? And that's what we have. By the way, dolphins have it too. Yeah, a lot of really advanced cetaceans, which are like your aquatic mammals, have really big brains. I don't know, if we can ever get to talk to them, we may figure out that we should have given them the reins long ago. The problem is that our communication between us and them is really limited. Octopuses have fantastic capacity as well, but their brain doesn't look anything like this. It's like two little blobs, and then the rest of the brain is spread out into all of their arms. Like a totally different evolutionary path for brain development with a lot of capacity. I don't understand what the hell's going on there, but if you guys ever learn it, come teach me. Okay, so let's talk about your whole brain, and we are going to start talking. First, we're going to go from the outside in. We're going to go from the places that are familiar to you, which is the cerebrum. We're going to start there, right? And even though it develops from the inside out, we're going to go from the outside in so that we can talk about stuff that you're actually interested in before you glaze over. Okay, so cerebral hemispheres first. Your cerebrum. I forgot to show you some cool stuff. Actually, I'll show you in a minute. So your cerebrum is divided into two halves, and I know you've heard this before: left and right cerebral hemispheres. Each cerebral hemisphere has some characteristics that you can see, right? It's got hills and valleys. Do you see the hills and valleys? The hills, and of course, why does it have hills and valleys? We already just talked about it. Yeah, surface area, exactly. So the hills and valleys are basically surface area. And why not just make your brain bigger? Well, because it still has to come out whole, right? Okay, so you've got to make more surface area without increasing the size of the brain, right? Okay, so the hills are called, singular is gyrus. So this is a gyrus, for instance. Plural is gyri. Nobody says it that way, but my neuroscientist friend who also speaks Latin told me that that's the way to say it. I feel pretentious as hell saying it that way, but I think it's right. Okay? Huh? Gyri. And then this is the one that sounds really stupid when you say it right. The valleys, yes? The valleys, singular is sulcus. Plural should be something related to sulc, but it's sulci. What? I tried not to believe her, but she knows more than I do, so I do believe her. Okay? Gyri and sulci. Okay? And so you see all these gyri and sulci, but then notice every once in a while, so you have a hill and a valley, but every once in a while you got a freaking Grand Canyon. That's called a fissure. You don't have very many of those. And where you have them, they have, what are those called? She's got it. Yeah, the fissure. It's a fissure, right? And then it has what in it? Remember?
The Venus.
He's got it. It's in there somewhere. Cranial dural septa. So this is a fissure. This is longitudinal fissure. You have another one that will have the lateral. Oh, where is it? Where's that stupid picture? Hold on. You're going to have another one. Oh, on this. I'll show you this picture. You're going to have another one that goes right there, right? A fissure that goes between the cerebrum and the cerebellum, and you'll have another one between the two halves of the cerebellum. So basically, you've got fissures wherever you had those cranial dural septas, right? Everything else is just gyri and sulci, which sounds weird. I acknowledge. Okay? And your labs instructor is totally going to tell you you're saying it wrong, and you can go, okay. Okay, so gyri, sulci, fissures. And then how is this related to the story that we told before about ventricles? Just to reinforce the stuff that we did last time. How are the lateral ventricles related to the cerebral hemispheres? Somebody give me the words that describes the relationship of the lateral ventricles to the cerebral hemispheres. A statement. You can do it. Go ahead, Jasmine. No? Doesn't want to do it. Go ahead. Who else?
Wouldn't it be something to do with the cerebral liquid?
Yeah, it's cerebrospinal fluid. Okay. So now we're going to put together all three of them. Okay. So the lateral ventricles are what? Within, yes, the cerebral hemispheres. Yes? That makes sense? Okay. And what is within the lateral ventricles? Cerebrospinal fluid. Yeah. Okay. Got it? Okay. Everybody good? So the lateral ventricles are inside of here. Just so you know, the cerebral hemispheres, if we look at them, they are gray matter on the outside and then white matter on the inside. Yes? Okay. That's what they're going to be like. So we're going to start to learn the portions of the cerebral hemisphere. And let's talk about that real quick before we get into the details. Okay. So the cerebral cortex, let's look at it. The cerebral cortex, guys, is the outermost portion of your cerebrum. It's the newest portion of your brain. And here's the deal. It's still true. The whole story is true. The oldest portion of your brain, both embryologically and evolutionarily, is the brainstem, right? And then the newest portion is your cerebrum. But the newest portion of the newest portion is the most anterior portion of your cerebrum. This portion of your cerebrum right up here is the very, very last portion to develop. As a matter of fact, in quite a few of you, it's not even done yet. Because the most anterior portion of the cerebral cortex is called the prefrontal cortex, as in the front of the front of the front of the front of the cortex, right? Most superficial, most anterior, right? The prefrontal cortex, we have found out within the last 10 years, does not complete its development until the ages of 22 to 25. So, a lot of you guys are still not baked yet. Okay. Now, why the hell is that? It gives you a lot of possibilities, a lot of neuroplasticity. And also, if I tell you what the prefrontal cortex does, as I tell you what the prefrontal cortex does, I want you to start to think about why you wouldn't want it to, for instance, be done by the time you were 15. So I want you to think about what it does and then try to put that story together, and then we'll come back to it. Okay? So we're going to be talking about the whole cerebral cortex. It is the newest portion of the human brain. The whole cerebral cortex is the newest portion. It's only 2 to 4 millimeters thick. It is where consciousness exists. Consciousness. Now, consciousness is a weird thing, and neurologists still argue about what the hell it is, right? But how many of you guys have dogs? Okay, so for those of you who have dogs, does your dog have some kind of consciousness. Yeah. Do they have desires? Do they have both sensory input and motor output? Do they have the capacity, as far as you can tell, right, for pleasure, displeasure, those kinds of things, right? Some of them even have anxiety, right? Okay, so consciousness, first of all, neuroscientists call consciousness the hard problem. This is what they call it, because they're like, what the hell is it? When did it come? Who has it? Who doesn't? But I think that if you really examine it, you will probably, maybe, come to the conclusion that a lot of people come to, which is that consciousness is a spectrum. It's not a bimodal distribution like we have it and nothing else does. Does that make sense to you? Like your dog has it perhaps more than a chicken? Yeah? I mean, I don't speak chicken, but I don't think a lot's going on there. And a chimpanzee perhaps has it more than your dog does. And you have it most of all, and maybe only because you made the measuring stick. I don't know. But regardless, it seems as if consciousness is a spectrum. And as far as we know, consciousness has something to do with the cerebral cortex. It exists in the cerebral cortex. How it exists in the cerebral cortex is the hard problem. Like, is that the side of consciousness? Is a certain thickness the side of consciousness? Everything below that, you're not conscious. Everything above it, you are conscious. We haven't come up with the answer to that. But we do know that in the absence of the cerebral cortex, you probably have nothing akin to consciousness. Okay? That makes sense? And the more cerebral cortex may be correlated with more consciousness, but let me show you something related to that in just a second. Okay, the other thing that happens in the cerebral cortex is that is the site of voluntary initiation of movement. It's where your voluntary initiation of movements. It's where all your higher thought functions go. And there's been a whole bunch of research about the cerebral cortex, and one of the main people who did it historically was called Brodmann. And you can read about Brodmann's areas in your book. So I want to show you two things. I want to show you that somebody made a space-filling model of how the cerebral cortex folds. I want to show you what the cerebral cortex looks like at 24 weeks. And then I want to talk about how we know a lot of what the cerebral cortex does. So first thing is that some brilliant person, and I think this was last year too, made a space-filling model that shows you the development and the folding of the cerebral cortex, which is just cool as hell. I love clever people. Right? So this is not cerebral cortex material, but they made a cerebral cortex that is a similar developmental speed and a similar texture to that exists in humans. And they allowed it to fold multiple different times until they came up with something that correlates to what we know about cerebral cortex development, which is pretty interesting. So you can see it go from very little cerebral cortex to a lot. This, again, happens mostly in the later stage of the second trimester and the third trimester. So although this makes people uncomfortable, I want to show you what the cerebral cortex looks like of a 24-week fetus. This is what the cerebral cortex looks like of a 24-week fetus. This obviously postmortem, right? Has to be. But it brings up some thoughts in some of us. Perhaps not in others, but some thoughts that I am not going to tell you what to do with, but some thoughts that are interesting to wrestle with. That this is the cerebral cortex of a 24-week fetus. It looks very little as far as folding capacity, like a goat has about the same as a rabbit, nowhere near as much as your dog, and certainly nowhere near as much as a fully formed human. By the time the baby is born, it has almost the same folding as you will have as an adult. But it all happens really late. 24 weeks. 24 weeks. So do with that what you will. Okay, so before we get into the cerebral cortex functionality, I want to talk a little bit about how we learned this stuff, because there's some dark history in here, and it's worth acknowledging that some of the history of the cerebral cortex and how we learned what the brain does is dark. So the cerebral cortex is divided into lobes, and what we have done is assigned functionality to a lot of different portions of the cerebral cortex, but there's kind of two different ways that we've learned a lot of this. Some of it's anecdotal, right? Like somebody sticks a fork in this part and they can't smell anything anymore, right? And then they're like, oh, that's the smell part. Or have you guys heard of Phineas Gage? No? I'll tell you the Phineas Gage story in just a second. basically he got a big old piece of metal stuck through his prefrontal cortex and started behaving like a whack job. People used to like him, hated him, and we learned a lot about prefrontal cortex function with that. Or somebody got a tumor and they lost this functionality or it changed, right? So all of that's kind of anecdotal. And then the dark history of the learning brain function has to do with the fact that we don't have a model organism that we can experiment on to learn very much about higher human functionalities. Does that make sense to you? We can tell about rat intelligence by how quickly they get through a maze, but we don't really know anything about rat emotions, right? And we don't know anything about rat personality. We don't know anything about higher level rat intellectual capacity because they're not a good model organism. And even chimpanzees, right? First of all, a lot of people have ethical consideration about experimenting on really, really advanced primates anyway. But even if that didn't exist, they also don't have the speech function to communicate with us what's going on. So we've got not a good model organism for higher functionality. And then in addition, I'm not going to let them experiment on me, are you? Right? Be like, I wonder what we could do that would make her more socially appropriate. I'm going to try to take that part out. I'm literally going to say no if you ask me, right? Okay? Right? I'm going to take that part out. So we can't do that because of human rights. Now, historically speaking, we have been less respectful of human rights than we are now, right? So it has happened in the past in which we have experimented on humans, and a lot of that information has ended up in your textbook. And a lot of that happened here in the United States, especially during the eugenics movement. I don't know if you guys know about the eugenics movement in the early 1900s. We considered certain classes and categories of people as slightly less than, and we did not feel any compunction about experimenting on them. So if, for instance, you were incarcerated or you were in a mental institution, we could take out a little chunk of your frontal lobe just to see how it changed your behavior. And we have done that a lot here in the United States. And then terribly, terribly uncomfortable to talk about, but one of Nazi Germany's biggest experimental scientists actually got that idea from us and did a whole bunch of experimentation on Jews and homosexuals and gypsies and all of the populations that were abused during World War II in Nazi Germany. So a lot of this information came from there. And it's worth acknowledging that this stuff doesn't exist in a vacuum, and also saying that as globally, we do have human rights expectations right now. And it's very difficult to get away with that stuff anymore. And the reason it is because you can't publish your data and actually get it into the scientific record and actually be a published, respected scientist unless I can track back to where you got it, right? And I can say it came from a source that was approved by this international human rights agreement. But historically speaking, that's not how we've done it. So let's talk about what we know about the brain. All right, so the brain is divided into lobes. This picture kind of shows them, but I think this actually shows it better. Or the cerebrum is divided into lobes. Here's the frontal lobe, which roughly correlates to the frontal bone, but it like goes back further than the frontal bone. Yeah, do you see that? And then of course this is the parietal lobe. Yeah, occipital lobe. And then temporal lobe. So super easy, right? Okay, but of course they're not really color-coded on the brain, so it's not quite as easy as you think. So I want to talk about the frontal lobe first, and we'll talk about its functionality. There's no way in hell I could teach you all of the functionality of any of the lobes, so I just sort of have to pick and choose things that make good stories or things that I think are going to be important to you later. So let's talk about the frontal lobe. Okay, so the frontal lobe anatomy. So let me tell you, and this is going to be hard right now, but easy later, like a lot of things. The frontal lobe ends at a specific sulcus, okay? This sulcus right here is called the central sulcus, and it's about right here on your head, okay? And it's actually not that hard to find because it's the only sulcus that if you look at an intact brain from a top view, superior view, you can see it going all the way across. Do you see it? That's the central sulcus, right? All of the rest of the sulci don't do that straight across like that. So do you see that central sulcus? Okay. Everything anterior to that is frontal lobe. Okay. How about lateral? If you're looking laterally, the frontal lobe ends at this guy right here, which is called the lateral sulcus, right? So everything anterior to the central sulcus and everything superior to the lateral sulcus is all frontal lobe. And again, your frontal lobe is the last portion of your brain to develop, evolutionarily and embryologically. And so it does some cool stuff that is really, really related to a lot of human functionality. So let's talk about the frontal lobe and learn some parts of the frontal lobe. So let's look at this picture from your textbook. Okay, I don't know why they didn't put it in here, but I think it's super important to talk about. The most anterior portion of your brain, right up here, is called the prefrontal cortex, right? So, as in more frontal than frontal, and the cortex. So I want to talk about what the prefrontal cortex does and what we've learned about it over the last few years. The prefrontal cortex is the portion that does not complete its development until 22 to 25, but it's different in the different sexes. Okay, so this portion of your brain is primarily related to planning, reasoning, judgment, personality, intelligence, social skills. So here's what this portion of your brain does. You have all of these antisocial thoughts every day. Everybody does. You may think you're alone, but it's just because people are lying to you and telling you that they're fantastic humans internally. But no, they want to flip people off, right? They think about punching people. They think about stealing things, and they don't. And the reason that they don't, and toddlers do, yes? You want that and you're a toddler, what do? You take it, right? Something pisses you off and you're a toddler, what do? You punch or you bite or you do whatever, right? And then the process of development allows us to take these drives that we have, that everybody has, and layer over them our prefrontal cortex activity. And our prefrontal cortex activity has the capacity to imagine the impact on our social situations. It's really, really important for appropriate social functioning. Yes? All of those drives that you have, you layer your prefrontal cortex over them, and then you behave differently. And that develops as childhood develops, and you can see it, right? Children actually behave differently as they get a little older. And you think it's just training, but it's also brain development, okay? And then it also allows you, and this is a big one, especially for those of us who are over 25 to think about. It also allows you to put off short-term desires for long-term goals. I want this right now, yeah? Okay, I want this right now, I could steal it, right? But I also would like to not be incarcerated, so I'm not gonna, right? Okay? Or whatever reasoning that you use, right? I think it might be a good idea to take crap, but then I think not, because I want to be a grown-ass woman with a job, right? Okay? So what it does is it allows you to put off short-term desires for long-term goals. And you have probably seen that develop in yourself. Have you not? As you age, even if you're not 25 yet. If you think back to some of the decisions you made when you were 17, your current self is like, WTF, past self? What was that all about? And it was your prefrontal cortex, among other things, right? So your prefrontal cortex, and this is interesting, your prefrontal cortex actually develops and matures later in one sex than the other. And I'll bet you know which one. And I'm not being sexist because there's an evolutionary value to that. So let's talk about it. Which sex do you think that it matures more slowly in? Males. And why? Is it just because I'm a female and I want to diss males? No. It's because there is an evolutionary value to males about being more experimental and less worried about consequences that lasts for longer in males than it does in females. Now, why the hell would that be true? Any idea? It's because of puberty and sexuality. Okay, so I want to think about the two sexes. And what, for instance, roaming far afield and maybe getting killed, but also maybe finding somebody new to have sex with, right? Is that particularly risky for a male compared to a female? How about sex? Is sex particularly risky for a male compared to a female mammal? It's not particularly risky, right? For a female mammal, it's resource intensive, it's physically dangerous, right? And it also reduces your mobility and it increases your dependence on other beings, right? It does, right? So it's really risky. Once you hit puberty, then a lot of times females' prefrontal cortex starts thinking about consequences more quickly than males start thinking about consequences. And not just male humans, male mammals. Male mammals tend to be more experimental for longer. And why is that? Well, evolutionarily, they can spread their seed as much as possible with very little extra risk, right, compared to a female. So in mammals, the experimental behavior tends to last longer in males than it does in females. And it's interesting to talk about because if you're a heterosexual woman, you are going to be dealing with a heterosexual, I hope, that's ideal, right? With a heterosexual male who may have things that are slightly different than you. Or if you're raising a son, right? You can't just tell them to behave like I did when I was 20. Because no, they're probably gonna have sex with her and also move to Egypt for 6 months without any money or being able to speak the language, right? And you basically have to say, I really, really hope that you survive this and I support you and here's my number and here's a credit card, right? And please come home and also stop doing that eventually because you're driving me crazy, right? But it is evolutionarily a pretty sound strategy, and it matures about age 25 in males. And in some of you, it hasn't matured yet. So for that reason, I have the unpopular perspective. I know it's the only one I've ever given you, that I don't think you should be able to get married before you're 25. I don't think you should be able to have a baby. If somebody asked me, I would say this stuff should be off by default. Just turn it off, right? I don't think you should be able to join the military or join a religion. None of it. Till you're baked, man. You got to get some baked going on. But nobody asked me. And you didn't ask me either, but I told you. Okay, so that's the prefrontal cortex. All right, so now let's get to some simpler functionality, which is kind of interesting. All right, so we're going to move back from the prefrontal cortex. So back here, do you see this gyrus that is just anterior to the central sulcus? Do you see that? Just anterior to the central sulcus. So listen to this, because this gets confusing. But if you listen when it's going through, you won't be as confused. Anatomically, that part is called the precentral gyrus. And that makes sense, yes? Because it's the gyrus that's previous to the central sulcus, yeah? Precentral gyrus, okay? But that doesn't describe what it does at all. So now I'm going to tell you its functional name. And its functional name is the primary motor cortex, and then I'm going to describe to you what the hell that means. So this portion of the brain, and I'll show it to you in detail in just a second, is in control of voluntary activity, motor activity of the other side of the body. So here's the deal. Here's my right hand. I'm doing this and I'm moving my arm. That is going on a stripe, being initiated from a stripe right here on the left side of my brain. Okay? I'll talk about why it crosses over in just a second. So this is called the primary motor cortex. And this one controls this half of my body, right? And this one controls this half of my body, right? Okay, so we have mapped out the primary motor cortex pretty well. You guys ready to see it? Okay, so I want you to stick with me and imagine this. So I'm going to take the primary motor cortex and I'm going to do a coronal section through it, right? And then look at where it's mapped. You with me? Okay, so this picture right here, orient yourself. So this is that big Grand Canyon in the middle, right? The fissure, longitudinal fissure, okay? This is the lateral portion of the brain, yeah? And this is the temporal lobe. Everybody kind of understand what we're talking about here? Lateral portion of the brain. So what they have done is they have mapped motor functionality on, let's say, the left side of the brain right here, okay, to what it controls on the right side. And now I not only know that this stripe right here is controlling my hand, I know that it is specifically this portion of the stripe that's controlling my hand. Now let's look at the real estate. Some places in your body get more real estate for motor control. Why? Yeah, more fine muscle control. So what gets the most real estate, guys? Lips, teeth, gum, and face and hands. Yeah? Do you have very much motor control over your freaking elbow? No, I don't even know. Like, I can have an itch there. I don't even know it's there. I don't even know how my elbow wiggles. Whatever. There's some elbow shit going on, but I have hardly any control over it at all. Right? That makes sense? And then you've got all this that goes down into the middle. Does that make sense? So if I am wiggling my left toes right now, it's because this portion in the middle right here of the right side of my brain is actually causing that to occur. So that is, again, let's reiterate, because I had the hardest time learning this. So if you are anything like me, you probably do too. Anatomically, what do you call this stripe? Precentral gyrus. Functionally, what do you describe it as doing? Primary motor cortex, which controls voluntary motor activity of the other side of the body. Okay, so everybody okay with that? Okay, so some brilliant person did this. They said, okay, what I'm going to do is I'm going to make a model, a sculpture of the body that has the portions of the body as big as the motor cortex is. Does that make sense? Like spatially, and then they made it, and this is it. I want this so badly. What?
I'm wondering what that is, like the entire class.
That is what it is. Yes, this is. It's pretty fantastic, right? I want this so badly, and I told my husband, he's like, "You're going to put more weird anatomy stuff on our mantle," and I was like, "If I can find where to buy it, I totally will." This is in a museum in, I can't remember, Frankfurt or something like that. Somebody made this. So basically, this is mapped to how much space you actually get. It's pretty damn cool to me. But then, you know, all right. So that's the primary motor cortex. For those of you who are curious as to why left side controls right side and right side controls left side, I don't know why it developed that way, but I know why it exists. The reason it exists is because the wiring during the whole folding process of the brain, this is white matter, right? The wiring at some point during the folding process of the brain crossed, yes? So that everything that came from this side goes to that side, and everything that came from this side goes to that side. Nobody really knows why it occurred, but it's as old as fish. So it's way older than humans. Like really, really old in the vertebrate line. I don't know what the hell happened. And you guys know with evolution, a lot of times it's like trying to reconstruct a crime scene. Sometimes you just don't have all of the information that you need. And people have told me why they think this happens, but none of their arguments are any better than any of their other arguments. So I think a good answer at this point is, I don't know. I don't know. And sometimes that's the only honest answer, right? I don't know why that happened, but it is. There it is right there. And it occurs, by the way, in the white matter of the medulla oblongata, and it's called the decussation of pyramids. We'll learn it later. But anyway, that's why that occurs. Okay, so let me tell you one last cool part, and I'm not going to make you learn any more of the frontal lobe, because I know that you're saturated. This portion right here, okay? That portion right there is usually in most people only on the left side. There's a bit of hemisphere specialization that goes on, and the left side is sometimes specialized for certain things, the right side is specialized for others. The left side, in most people, about 80% of people, is where most of your speech is specialized, okay? And this is the first speech functionality that we're going to learn. This little area right here, do you see that it's just anterior to the precentral gyrus, right? And it's just superior to the lateral sulcus, right? So you can find it, right? So let me show you a different way. Let me show you here, okay? So it's called Broca's area. Where are you, Broca's area? Let me just show you here. So can you see it? Right? There's Broca's area because this is the central sulcus, right? This is the precentral gyrus. That's Broca's area. It's not very big. It's pretty damn important. And we know it's pretty damn important because of what happens when it gets broken. So let me talk to you about what Broca's area is, and then I have a video to show you guys about it. Broca's area is often called the motor speech area. And there are other speech areas to listen to what this one is about. This one is about... I'm thinking of an apple, and I need to communicate apple to you. And what I have to do is to produce some motor activity that either writes the word apple, right, or speaks the word apple. It doesn't matter what language, though, right? Because that's all just symbology that either we share or don't share. Does that make sense? Okay. So this is the motor speech area. If it's broken, it doesn't mean that I don't look at an apple and know that it's a thing to eat. No. It means that I cannot produce the patterns that will allow you to understand the image that's in my head. Does that make sense? So it's motor speech. Good? Okay, you got it? So the motor speech area is producing written and spoken language. Interestingly, it doesn't matter how you're producing the symbols. You need to be able to translate an idea into symbols. And isn't that what language is? Translating an idea into symbols and then using a similar symbolism so that he could understand what I was talking about? Okay, so what happens if somebody has a stroke or a damage or a TBI, traumatic brain injury, that damages that part? Well, I'll show you because this is a pretty famous case. Where are you? Okay, so her name is Sarah, And Sarah has what we call Broca's aphasia, which is, okay, hold on just a second. She has stroke. She's much better now. And she had a stroke that impacted her Broca's area, among other areas, and you'll sort of be able to see as she's talking. And you will see how that manifests, and hopefully it will help reinforce to you what Broca's area does. Okay.
Okay. So what's your name?
Scott. Oh, no. Sarah Scott.
That's right. And how old are you?
I can't.
Try.
I can't.
You're 19. 19. Yeah. And what happened to you?
Stroke.
You had a stroke last year.
Yes.
And what happened? Can you remember what happened?
School and English class,
Okay.
and I book and I read it aloud, but I can't because stroke, and so I turn and also
You
it's
see which arm?
the same as the same of kind of thing as you know,
Pins and needles.
yeah,
like pins
the
and
same, and also farmers, but
needles. Leg. Leg.
yeah.
And what happened after the stroke with your speech?
But I can't.
You have speech problems.
Yeah.
Do you know what that's called?
Yeah. Aphasia.
Yeah, good, good. I haven't heard you say the word before.
I write it down. Because,
And that helps.
yeah.
Okay. So you are going to go to university. What are you doing now?
But
What do you do now? Every day?
I'll speech then the speech.
Therapy.
Yeah.
Okay, so that's Sarah. Sarah has several other videos because this was in 2009. She's improved really, really dramatically since then. She has one from 2 years after this and one from 4 years, and I think there's one from last year or the year before. And this relates to Riley's question. She's not getting that portion of her brain back that had the stroke, but because of the neuroplasticity and the fact that our brain actually has the capacity to do more than it's doing, what happened is a different portion of her brain actually took on this functionality. So our cerebral cortex is really like this just general processor that has the capacity to take in and process, integrate a whole bunch of different kinds of information. And so that's what happened with her. So that's Sarah. Do you understand what's happening here? So you can see that she's not having trouble understanding the nature of the conversation, but having trouble producing the actual output from the conversation. And the interesting thing, well, there's a bunch of interesting things about Broca's aphasia. With Broca's aphasia, sometimes you lose speech, and sometimes you lose writing, and sometimes you lose both. And one of my students told me in the last class that her grandpa had a stroke, and his manifested. He's Dutch, but he had been in the United States for a really long time. He lost all of his English speech and could only speak Dutch. And none of his family spoke Dutch, so they actually needed a translator to speak to him. So he lost all of like one portion of his translating, not the other one. I don't know whether that's like how big the stroke is or anything. I don't even know if people know that, but I don't know that. So anyway, that's Broca's aphasia. And then I'll tell you one more thing about.
I have a question.
Go ahead, I'll try.
Can this happen to a child? Like, can you be born without?
Oh yeah, absolutely. Yeah, you can be born with like loss of functionality in any portion of your brain.
Children who have
we're about to talk about autism, and I won't have any answers for you, but I'll have a little bit of clarification about what we know about it and don't know about it. Also, let me tell you that this is a decent resource before I talk about autism in just a teeny bit. I'm absolutely no expert in autism. Autism is at least 6 different portions of the brain. But there are some things we know and some things that I can tell you. And also, here's a fantastic resource because I want to show you this. Where is it? Okay, so if you go to this same site, and the reason that I'm interested in this, by the way, is not just because I'm interested in neuroscience, but autism is really, really common in my husband's family. His brother is autistic. He's got an uncle that's autistic, and 2 of the 5 grandchildren are on the spectrum as well. So it's really, really common in my family, and so I've tried to educate myself as well as possible. So if instead of going to this page, when you get to G2C online, this one, you go up here, you can click on a disorder, right? Which I realize that some people want to recategorize autism from just being differently neuro. And you can look at so much about what's known with autism. For instance, if I want to know the neuropathology of autism or the role of an amino acid called GABA or, autism candidate genes, I can click on this. And one of the cool things is that you can also see an interview from a scientist who did the research on almost every single one of them. So how's the cerebellum, neuropathology, let's see, Fragile X syndrome. So this is one of the scientists who did the research. So not only can you read, but you can also hear her talking about her research with autism, which is not just true for autism, but also bipolar disorder and a bunch of different, they don't have everything in there, but they've got a hell of a lot. And if you're only interested in the genes, you can say, I'm only interested in the genes, only interested in biochemistry, brain anatomy, cognition, environment, all that kind of stuff. So you can go bonkers on that page if you want. I've spent hours and hours on different disorders there. Okay, so I want to talk about autism a little bit, but I want to talk, before I talk about autism, I want to talk about a couple of other things that are in the frontal lobe, although I'm not going to make you know exactly where they are because we don't know exactly where they are. Some other frontal lobe functions, we'll talk about that, and then we'll talk about Phineas Gage and imitation empathy and autism. Okay, so one of the things that the frontal lobe does for you is allows for a certain type of learning. And you've been a kid or you've had a kid or whatever, and you know that a lot of learning is imitation, yes? And so you do something and your kid does it, right? And your kid does it over and over again, and it's just freaking adorable the first time they do it, right? Little baby has no capacity to eat with a spoon, but they see you eating with the spoon and they start doing this, right? And it's just the cutest damn thing ever. And you laugh and they laugh and all that kind of stuff. So a lot of learning is imitation, right? And other mammals do it too. They just mirror what mom or dad does. And then that goes on primarily in the frontal lobe. But then interestingly, empathy or compassion, or however you want it, and they're slightly different, has to do with emotional mimicry. So long before a kid develops what's called theory of mind. Do you guys know what theory of mind is? Theory of mind is me being able to say that when Lauren experiences something, her emotions in that situation are probably similar to mine. So that, for instance, if she falls down, I say, oh, are you okay? Right? And I maybe try to help her up. That doesn't develop until really, really late. And initially you really think that you're just an autonomous being and nothing else exists that's anything like you as far as childhood. But you start to develop empathy primarily by mirroring the emotions that other people show you, right? So they experience something and they laugh. And long before a baby knows something's funny, you laugh, they laugh, right? You cry. Did anybody have a baby that cried when they cried? Yeah. My son used to get super duper sad whenever I was sad, super duper sad. And it was just the cutest thing ever. And it was just this. It's just emotional mimicry that leads ultimately to empathy and compassion. So I want to talk about autism related to that. So a lot of times people who have autism or are on the spectrum have difficulty with that emotional mimicry. Okay. So my brother-in-law, Rick. My husband's name is Mike. His brother's Rick. Rick is autistic. Rick is 2 years older than I am. And he is a very, very compassionate and empathetic person. But he didn't learn that by all the nonverbal cues that we do. What had to happen during his therapy as he was growing up is we had to say, hey, Rick, if you want somebody to know that you care about them, one way to do that would be to remember their birthday and anniversary and remember something from the last conversation that you had with them and bring it up the next time you see them. And so you're like explicitly teaching the things that are usually implicitly taught. Does that make sense? And what ends up happening is Rick thinks, I do care about this person, and I do want you to know that I care about you. So I am absolutely going to remember that. And guess what? Rick remembers him better than anybody else in the family. He's like nicer than the rest of us by far, right? Super nice. So what happens sometimes is people who assume that other people who are on the spectrum lack this emotion and empathy and compassion, but that's not the case. What they lack is the ability to learn it non-verbally without and not all of them, but Rick did, without explicitly saying cause, effect, cause, effect, cause, effect, and then reinforcing that with cause, effect, right? So when Rick calls you, you say, thank you for calling me, Rick, and then it reinforces it. And then he is fantastic at that. But it's a different development. So we do know that autism is a whole bunch of different places in the brain, but in one of the ways it's actually in the prefrontal cortex. But you can look at the research that's in there and it'll tell you all of that, the kind of stuff that we know about autism. Okay, I'll talk about Phineas Gage next time. So note to self, I know you guys won't do this until the end, until after you have your test. So a couple other things. Your test does not cover the brain. Can you see why I didn't put the brain on this test? Because it won't stick very quickly. You're going to have to kind of piece it out slowly. So I usually try very hard to make the most intense stuff at the beginning of a unit and not at the end of the unit. So you got time, just like all those muscle attachments and stuff. Your test covers, again, just to reiterate, muscle and all the muscles you learned in lab. Okay? How will I reinforce that? Instead of pointing to a muscle like they did in lab, I will ask it backwards. I will say, here's the origin, here's the insertion, here's the action, name the muscle. Right? Does that make sense? And hopefully that will just allow you to hold on to the stuff that you already practiced and apply it to lecture. And then I'll do muscle mechanics too, the muscle logic stuff that we've talked about before. And then the microscopic muscle stuff, good, right? Skin. And then nervous system 1, which was primarily all of that terminology, right? CNS, PNS, autonomic, afferent, efferent, all of that stuff. And then the difference between nerves and neurons and all the different kinds of glial cells. So those 3 sets of notes. Probably less skin than other things, right? Because the other sets of notes were longer. I usually look at how long they are and how much terminology is in there. And if I have time, I will post another quiz for you. And I will put a notification on Canvas. But I'm not going to promise you that I'm going to do that. If I find all the questions and can stick them together while I'm having a glass of champagne, then I will post them. Okay? All right. Have a lovely weekend.
Yeah. Okay, you just brought it up for me, but
Okay.
I'll let you finish saving.
Oh, yeah, let me save because I'll forget.
I